Good afternoon, grade 8. How is going? I hope all of you are doing well. This is Miss Christine, science teacher at Hussein International School. And let's revise again the steps of meiosis. So here we go. Okay. Okay, so we talked last session, the steps of meiosis, and we'll revise it again. So at the end of the session, we'll be able to know the steps of meiosis and how the genetic variation happens between people by crossing over and independent assortment. Okay, so let's take each stage and revise it again. So as you know that we start okay, so prophase. Okay, we have two stages for meiosis, meiosis 1 and meiosis 2. And in each stage, we have prophase, metaphase, anaphase, and telophase. And then cytokinesis to separate the two cells, okay, from each other. So let's start with prophase. As you can see here in this picture, prophase, the nuclear membrane starts to disappear and the two centrioles come to each side, okay, to each pole, and produce a spindle fiber which attach to the homologous chromosomes. Here we can see two chromosomes, two homologous chromosomes after crossing over here. And we said that variation between human due to this crossing over. So we can produce new chromosomes with new genes. Okay, so this is step number 1, prophase 1. And then we have metaphase, metaphase 1 in its shortest phase, tedra align on the metaphase plates at the center, and independent assortment occurs. So what the meaning of independent assortment occurs means the arrangement or the lineup of chromosomes happens independent, okay? So the orientation of homologous pair to poles is random. Number 2, there is variation. Number 3, we have formula to n, like as we watched in the video. We have 2n and n maybe 4, maybe 2, and all of these can reach us to 1 million variation. And here the picture for metaphase 1. So here the homologous chromosomes line up at the equator of the cell, and it's called metaphase plates. Okay, so maybe arranged like this way, or maybe arranged like this way. You can see here, look, okay, these chromosomes, faint blue, and here, dark blue, okay? So we have variation in the arrangement of chromosomes. Then, anaphase 1. Anaphase, which means the homologous chromosomes start to separate and move toward the poles of the cell. And sister chromatids, in this case, remains attached at their centromeres. But anaphase 2, in meiosis 2, they will be separated, the chromatids will be separated, okay? But here the chromosome at all will be separated, okay? With the new crossing over. So anaphase 1, here, two chromatids attached with centromere, and the two chromatids attached to centromere go to different poles. So during telophase, each pole now has haploid set of chromosomes because have the half number, okay? And then cytokinesis occurs and two haploid daughter cells are formed. We start with 4 chromosomes, will be 2 in each one. We have picture here. Yes, we have 4 chromosomes at starting cell, and then after anaphase, 2 chromosomes will go to each pole, and then will produce haploid cell. Each cell has just 2 chromosomes, okay? So the product of meiosis 1 will produce 2 cells with the half number of chromosomes. If we said that we'll start with 20 chromosomes, so at the first meiosis, we'll give each cell with 10 chromosomes only. Then we enter meiosis 2. And in this case, in this meiosis 2, no interphase 2. Interphase 1 happens before meiosis 1 to start to prepare the DNA, prepare the organelles, duplicate each organelle inside the cell. But here, no interphase 2, and or very short, it's very short, no more DNA replication, enough. And remember, meiosis 2 is similar to mitosis. So mitosis happens during meiosis 2 only. Why? Because we'll start with haploid cell and end with 2 haploid cells. So the same number of chromosomes. Okay, so let's start meiosis 2 with prophase 2. Same as prophase in mitosis. Okay, here we have 2 chromosomes align up, and centriole send spindle fiber and attach to each chromosome in the centromere. So then will be a lineup and forming metaphase plates at metaphase 2. Then anaphase 2, like in mitosis, each chromatids will be separated to each pole, to one pole, with the half number of chromatids. And telophase 2, same as telophase in mitosis 2, nuclei form and cytokinesis occurs. As we said before, that all things which disappeared in the prophase will reappeared again in telophase, okay? So 4 haploid daughter cells produced. Here we can see the process again. So we have 2 cells after meiosis 1, and each cell will produce 2 cells. So we have here 4 haploid cells. So we finished the steps, all the steps. And we have question here. So let's share. Yes. Okay. So we have quiz here. Number 1, a cell is going through meiosis. The sister chromatids are lined up on the metaphase plates. What phase of mitosis is this? Sister chromatids are aligned up. So which phase? Metaphase 1, prophase 2, metaphase 2. So which phase? Who knows the answer? Raise your hand. If you know, raise your hand. Who knows the phase in which metaphase plate forms and the sister chromatids are aligned up? Yes. Hala?
Can you hear me?
Yeah, Hala. Yeah, you know
So I
answer?
think it's metaphase 1.
Metaphase 1. So let's see the answer. Be with me. No, the correct answer is metaphase 2. Why? Because it's written here the sister chromatids, not the homologous chromosomes. The homologous chromosomes align up in metaphase 1. But in metaphase 2, the sister chromatids align up, okay? This is the difference between metaphase 1 and 2.
Okay.
Thank you so much, Hala. So let's read that full answer here. In the metaphase 2, in the metaphase 1, the homologous chromosomes are aligned up on the metaphase plate. This is an extremely important difference between the two. Metaphase 2 is just like mitosis, while metaphase 1 results in the reduction of ploids, okay, of chromosomes. Okay, let's take number 2. Number 2, an adult organism has 60 chromosomes. or 30 homologous chromosomes, because as you know that 46 chromosomes arranged in pair, arranged in homologous chromosomes, as we talked before. And 30 are maternally derived, 30 are paternally derived. 30 from mother and 30 from father. How many chromosomes are in each cell after mitosis? After mitosis. Again, read it again. Adult has 60 chromosomes or 30. Okay, so we'll start with 30 chromosomes. After mitosis, so who knows? Rumana? Fatma? Okay, Fatma.
Miss, can you again, sorry?
Read it again. Here we'll start with,
Yeah, please.
the cell here will start with 60 chromosomes. So after mitosis, the cell will have how many chromosomes? Mitosis, not meiosis.
It will have the half number, which is 30.
30?
Yeah, 30.
Okay, let's see. So the answer is 1, not 30. Because we said in mitosis, the produced cells will have the same number of chromosomes. We'll start with
In
2N.
mitosis.
Yes, I said mitosis. Focus. I said
Sorry, I
mitosis
heard
so
it
many times. Okay, so mitosis will start with 2N and end with 2 cells with the same number of chromosomes. So it will be 60 chromosomes.
Okay. Okay. Thank you.
Welcome. I think it's something tricky. Okay, so mitosis produces the same number of chromosomes. Essentially, mitosis produces an exact clone of the parent cell. A clone means like shape, okay? Okay, we have number 3 here. Last one. An adult organism has 60 chromosomes or 30 homologous pairs of chromosomes. Okay, and here he wants after meiosis. So the number of chromosomes are in each cell after meiosis. Okay, how many? It's easy now. After meiosis, not after meiosis, mitosis. Perry? Yes, Perry.
30 chromosomes.
Yes, 30 chromosomes will be the half number. Thank you so much. Let's see the full answer. Okay. A is correct. 30. Yes. Why? Too much answer. So, because meiosis reduces the ploidy of each cell, reduces the number of chromosomes. Effectively, this means that only one copy of each chromosome, either the maternal or parental copy, will be returned in each cell. The homologous copies of each chromosomes are stored on the metaphase plate in metaphase. He describes what happens in metaphase 1 and 2. Okay, so the correct answer will be 30 because meiosis produces 4 haploid cells with half number. Okay, we don't have more. So if you have any question now, you can ask me. Fatma? Yes? Okay. You can write or you can ask me. Yes, Juman.
Miss, I'm sorry, I have a question.
Yeah, sure.
Miss, but I don't get it. Didn't you say when it's meiosis, the like the number, like there are 2 new cells, like they come out 4? So why is it
Meiosis
still
or
30?
wait, sorry. Meiosis or mitosis? T or
No, meiosis.
meiosis. We said we'll start
Yeah.
the cell. We will start with one cell, parent cell, okay?
Mhm.
And this cell with the diploid number 2n, okay? Let's start with 40 chromosomes or 46 chromosomes, okay? So 46 chromosomes in one cell will give, after the end of meiosis, will give 4 cells. Each one have the half number, 23 chromosomes,
Yes.
okay? So here, he started the cell with 60 chromosomes, okay? So each cell will take the half number of chromosomes will be 30. And no homologous chromosomes. Why no homologous chromosomes? Because we said that we will have just one copy of chromosomes. 6 cells, sperm or ova or egg, okay, has
Yes.
one copy of chromosomes, no homologous chromosomes, because it's 23, not 46. But he changed the number. It's written here 60 chromosomes, okay? So the half number will be 30.
Yes.
30 with no homologous chromosomes. Clear now?
Okay. Yeah.
Okay,
Okay, thank you.
welcome. So, do you have any question? So let's read back. I want now to listen to the video or watch the video again to summarize the whole process of meiosis and to be more clear. So one more time, please. Save, once, okay.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells.
So here, guys, we start with gonads, okay, which is female, like here, female, with diploid numbers. The cell start with 46 chromosomes to N. So at the end of meiosis, will produce egg with the haploid number, which is N 23 chromosomes. Also in male, will start with germline cells, or it's called the organ, okay, the male sex organ, with cells 46 chromosomes to N. So at the end of meiosis will be the half number, which is in the form of sperm, haploid N equals 23 chromosomes, okay? So we start with somatic cell to bring 6 cell. Let's continue.
With half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis 1 and meiosis 2.
Here, guys, as you can see here in this picture, meiosis 1 and then meiosis 2. We start with just one cell. Here, we see with arrow. We start with one cell, which is somatic cell, with the diploid number, 46 chromosomes. And then after meiosis 1, we'll give 2 cells with the haploid number, N and N. And then each cell will give 2 cells, so the yield will be 6 cells with the half number, which is N, 23 chromosomes. Okay, and now you compare between meiosis and mitosis. Meiosis ends with 2 cells with the same number of chromosomes, so 46 chromosomes somatic cell will give 46 chromosomes somatic cells too, okay?
Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase 1 of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase 1, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called
Here, you can be focused, okay? The homologous chromosomes, they are similar in size, okay? But not have the same genes, okay? Maybe this gene recessive or dominant, okay? Maybe it will appear or not in the first generation or second generation, but they are similar, okay? So it's called homologous chromosomes. Now we will discuss or explain how crossing over occurs, and this crossing over causes variation or diversity in generations, okay? So let's listen.
crossing over can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase 1 concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move,
What may have a question? Yes? Yes,
Miss, just to ask, is the crossing over also happens in mitosis?
no. Because in mitosis, there is no homologous chromosomes, okay, separated. And in mitosis, the aim of mitosis to produce the same typical cells, okay, for repairing cells or compensate the dead cells, okay? But crossing over happens during meiosis only to give variety of genes.
Okay.
Yeah? Okay,
Thank you.
welcome. So let's continue.
the centrioles extend spindle fibers forming the meiotic spindle. In prometaphase 1, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes
Look here. Okay, we have here this picture for mitosis. We can see just one chromosome, not homologous chromosomes, okay? Just one chromosome consists of 2 sister chromatids, okay? And here in meiosis, we have homologous chromosomes, 2 chromosomes. So, because the 2 homologous chromosomes arranged or align up at the center of the cell, so making crossing over. But here we have just one chromosome. How crossing over will be happening? Okay?
remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase 1, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes separate and move toward opposite poles. Meiosis 1 ends with telophase 1, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the
So here in this picture, you can see like one chromosome align up, okay? So it's like mitosis. In this case, like mitosis. No homologous chromosomes. Just one copy of chromosome here, okay? So we'll complete like mitosis.
metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase II, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase I, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase I, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes.
So we have 2 causes for the variation of offspring, okay? Number 1, crossing over. Number 2, like it's written here, independent assortment, means the independent assortment or arrangement of homologous chromosomes here at the center of the cell, okay? Okay, we have just 2 minutes. If you have any question, you can ask me now. And so, Fatma? Yes, Fatma?
Miss, can you please say the difference between haploid and diploid again? Sorry.
Haploid means that the half number of chromosomes, N, okay?
Okay.
Diploid,
And diploid
it comes from di. Di means 2, okay? So diploid means 2, which is 2N, or the full number of chromosomes. So if we start with 46 chromosomes, it will be diploid number, okay?
Yeah.
Because it's 2N, and the haploid number will be 23.
Yeah, that one.
Okay, I think now we covered all parts or everything in meiosis in details. So, do you have any question before? Okay, so I want to say thank you so much for watching, and if you have any question, you can ask me. Okay, and enjoy this video. Bye bye. Okay.
Nope.